So this next module is going to be on the cells of the lymph and immune system, and we'll start with just one slide on the lymphatic cells right now, and then we'll revisit it a little bit later. The cells in your lymph fluid include NK cells, which are natural killer cells, T cells, and B cells. We'll talk about NK cells a little more later also, but for right now, what I need you to know is that NK cells are very non-specific immune surveillance cells. They simply swim around your lymphatic fluid looking for anything they don't recognize. And if they find something they don't recognize, they do phagocytosis and engulf it. The T and B cells, however, are for your specific immune system, and they are a big class of cells that you have to turn on to fight against specific antigens on bacteria. So I'm hoping you remember this, but on cells, any cell in your body, you're going to have a signaling molecule called an antigen. And this antigen tells everyone around it who it is. So if this is your bacteria, the bacteria is going to have an antigen that says, hey guys, I'm a bacteria. So T cells and B cells, when you activate these guys, they go out into your body and they look for specific antigens to kill. Now, this happens when your body's immune system can't fight off the bacteria initially, and that bacterial or viral sickness gets much larger. Then you bring in the head honchos to go after a specific antigen, and they do a very good job of it. They're usually able to fight off all bacteria or viruses with this antigen within 1 to 3 weeks. So the T cells, these are considered T because they mature in the thymus. The B cells are called B because they mature in the bone marrow. Now, the cool thing about B cells is, let's say you have a B cell here, and the B cell encounters a bacteria it doesn't recognize. Well, the B cell itself doesn't attack bacteria. The B cell turns into this kind of gloopy-shaped plasma cell. So, when a B cell gets activated, it converts into a plasma cell, and the job of the plasma cell is to make thousands upon thousands of these structures called antibodies. Antibodies come and bind to foreign antigens. And when antibodies and antigens bind together, they kind of set off an alarm system to tell the rest of your body, hey, we created an antibody-antigen complex. That means that this cell is bad. Whenever an antibody binds to an antigen, that usually indicates an immune reaction, and it sends a signal to the rest of your immune system to say, hey, come and get this bacteria and engulf it. So usually at this point, and we'll talk about this more later, a really large macrophage will come and it'll say, hey, I see this antigen-antibody complex, I'm going to eat it. So it eats the whole bacteria. So it's kind of a cool thing that B cells can do. They can get activated to turn into a plasma cell. The plasma cell makes antibodies. Antibodies bind to antigens. And when you have an antibody-antigen complex, it sets off an alarm that calls over macrophages to eat and engulf that foreign cell. So there are 3 lines of defense that are part of your immune system. The first line of defense and second line of defense are both considered non-specific. These 2 lines of defense will fight against any bacteria or pathogen that they identify, and they don't go to identify specific bacteria or antigen, they just identify anything they don't recognize. So what that means is the first line of defense includes things like your skin, mucous membranes, or any external barrier that creates a physical separation between your internal environment as well as the external environment. The second line of defense is again another non-specific defense, and this one is made up of actual immune cells like leukocytes, macrophages, antimicrobial proteins, processes like inflammation and fever, all of which do fight against bacteria, but not against specific bacteria. The third line of defense, however, is your specific immune system. We call this your immunity. The immunity is made up of T cells and B cells that are activated to go into your body, hunt for, and kill specific bacteria that present a specific antigen. I guess a key word that has to do with third line of defense is the word antigen. There has to be a specific antigen we're fighting against, and a specific antigen we can turn into memory of your immune system so that you don't get infected with that same antigen again in the future. Now, going into a little bit more detail, the first line of defense, external barriers like skin or mucus. Your skin has two characteristics about it that actually help make it a line of defense. And the first one is that it secretes something called human defensin. And human defensin is a naturally made antibiotic on your skin that kills bacteria by punching holes in the cell membrane. The second thing about skin is that it sweats. Of course, you all know your skin sweats and secretes water, salt, but that sweat is considered to be slightly acidic. And that's because there's a little bit of lactic acid in there. That lactic acid is something that can kill bacteria. Now, internally, you've got mucous membranes, digestive system, reproductive system, urinary, respiratory. All these are lined with mucus. And what's cool about mucus is that it has lysozymes in it. And lysozymes are there to destroy bacterial cell walls. So lysozymes is that organelle that we've seen inside cells before, but they are also in mucous membranes. So again, defining what's nonspecific, like the skin or mucous membranes, versus what is specific, like T cells and B cells. The nonspecific immune systems is the first and second lines, and these nonspecific resistance of your body include the skin, the mucous membrane, and all the cells that we listed. The immunity, that specific resistance, again, key word with this one is antigen. These guys are immune system cells that respond to the presence of specific antigens. Now, the nonspecific immune system works a lot faster, but it's less effective. The immunity or specific resistance takes a bit of time to build up its response, but it works very, very well. So we're going to now look at a breakdown of all those cells, and I'm going to kind of give you a big picture, and then we're going to go into the individual cells and their functions. Your immune cells all come from stem cells. So these blood stem cells in your bone marrow will differentiate into two groups, either myeloid stem cells or lymphoid stem cells. The lymphoid stem cells, they have the prefix lymph in them, which means they're going to create cells that are found in your lymphatic fluid. So the lymphoid stem cells will eventually differentiate into three cells: B lymphocytes, T lymphocytes, and NK cells. We already went over these three. These three cells are considered lymphocytes because they live in your lymphatic system. The myeloid stem cell line will differentiate into red blood cells, platelets that allow for clotting, and these three cells called granulocytes: Eosinophils, Basophils, and Neutrophils. These three are found in the blood. These three are found in the lymph. We call these lymphocytes. We call these granulocytes. But all six of them together are grouped as leukocytes. Leukocyte just means white blood cell. All six of these are types of white blood cells. So we're going to talk about all six of these. I'm going to give you the names. I'm going to give you the functions. And then we're going to really dive into the specific immune system after that. So again, your job is to learn the name of the cell and the function of the cell. We got neutrophils, eosinophils, and basophils. These three are going to be in the blood. We've got monocytes, which eventually turn into macrophages, and T and B cells as well. And these ones down here are located in the lymph. So first one, a neutrophil. Neutrophils are going to be cells inside of your blood And when they come into contact with bacteria or cells they don't recognize, they do a few things. They can do phagocytosis and digestion of that bacteria, or they can do what's called degranulation. Degranulation is when the lysosomes, that organelle inside of this neutrophil, discharges toxins out to the environment around it. That process is termed respiratory burst. Respiratory burst is when the neutrophil itself discharges toxic chemicals like oxygen radicals and let's just say hydrogen peroxide into the area around it. Once it does that, that area is now called a killing zone. Any cell within the vicinity of that killing zone is going to die, whether that's a bacteria or whether that's one of your natural cells. So neutrophils, the best way for them to function is to find a colony of bacteria or virus, go into the middle of them, do their respiratory burst, create that killing zone to be able to kill the pathogens that are in that area. Once it kills them, it can then do that Pac-Man-like function and do phagocytosis and eat them. The second type of leukocyte in your blood is called an eosinophil. Eosinophils do the same function as neutrophils do, where they can do respiratory burst. That's this second bullet point. And they can secrete these toxins to kill their surrounding cells. They are also able to guard against allergens, and they're able to do phagocytosis of antigen-antibody complexes. And just to remind you, here's your bacteria, here is your antigen. A B cell will turn into a plasma cell, and the plasma cell makes antibodies. And if you recall, those antibodies create a signal. And that signal will call for other immune cells, like eosinophils, to come and do phagocytosis of this. The third leukocyte that's found in your blood is called a basophil. Now, unlike neutrophils and eosinophils, basophils do not do phagocytosis. They do not attack any pathogens themselves. But these guys act more as messenger cells. When a basophil comes into contact with a potential bacteria, it secretes 3 chemicals. The job of these 3 chemicals really is to help generate an immune response. And the first chemical is called a leukotriene. The first step in generating an immune response is to call over cells that are going to generate the immune response. So the basophil will secrete leukotrienes. This chemical will then activate and call over neutrophils and eosinophils, the 2 that we just went over, to come and create a killing zone and do phagocytosis. Basophils will also secrete histamines. This is a chemical that acts as a blood vessel dilator. And if you dilate blood vessels, you're going to allow more blood flow and more white blood cells into an area of infection. And the last one is heparin. Heparin prevents blood clotting. Again, if you have an infection somewhere, you don't want to clot the blood going into that area because you want to make it as easily accessible to white blood cells as possible so they can kill the bacteria and clear it from the tissue. Then you got monocytes. Monocytes are large cells from the blood that eventually transform into macrophages, and these guys do phagocytosis. There are 2 types of macrophages, wandering and fixed. Wandering macrophages, we touched on this I think a few slides ago, maybe we didn't, but wandering macrophages are the types of macrophages that are able to walk around your body, and they have these like legs and branches that can extend out pretty far. And these guys go around your body actively looking for just pathogens in general, not a specific pathogen, just a general pathogen that has a foreign antigen that we don't recognize. It seeks it. Once it finds it, it does phagocytosis. And then you got fixed macrophages. These guys do not walk around. They are stationary. They stay where they are rooted, and these guys will only do phagocytosis of pathogens that come to them. For example, microglia are in your central nervous system. Alveolar macrophages are in the alveoli of the lungs. Hepatic macrophages are in the liver. These fixed macrophages are in the tissues of those organs and they do not move around. The only time they do phagocytosis is if a bacteria comes into its vicinity on its own. And the last piece here, you've got your lymphocytes, the T cells, the B cells, and the NK cells. If you notice, the T cells are much more numerous than the other two put together. Next to T cell, I'd like you to write attack cell. A T cell is an attack cell. This is a cell that attacks bacteria directly. Where the B cell, if you recall, a B cell doesn't attack bacteria. When you activate a B cell, it turns it into a plasma cell. The plasma cell produces antibodies. Antibodies bind to antigens, and that antibody-antigen complex creates a signal that calls for other immune cells to digest that cell. The last one is an NK cell. We only have 5% of these. These guys act like macrophages themselves in the lymphatic system. They are large, they swim around, and whatever they don't recognize, they're going to do that Pac-Man-like move and engulf it and digest it. So this is where we start talking about the specific immune response. We're going to go into a little bit about T cells and B cells and how they become mature. So T cells are produced in the bone marrow. They leave the bone marrow and they go to the thymus, hence T cells. In the thymus is where your T cells mature into adult T cells that actually function properly. In the thymus, you make a hormone called thymosin. This is the only function of the thymus, is to make thymosin to use to help T cells mature. This hormone stimulates T cells to develop surface antigen receptors. This is going to be something that looks like this. Here's your T cell. In the presence of thymosin, it allows us to develop this receptor that sits on the membrane of T cells, and I'm going to call that my T cell. And these receptors are there just in case, let's say you have a bacteria, or let's just call this an antigen-presenting cell. We'll talk about that in just a moment. Let's say this is an antigen-presenting cell. This is a kind of cell that presents antigens. So here's the antigen here. When this antigen comes and binds to this surface antigen receptor, that is one way to activate a T cell. Without these receptors, a T cell is useless because a T cell won't be able to recognize any foreign antigens. So thymosin is the hormone that stimulates T cells to develop these receptors on its surface to prepare itself to go and monitor your body. We now have to make sure the T cells are what we call immunocompetent. We have to make sure that these receptors only react to foreign antigens. It would be very bad if these T cell receptors would bind to your own antigens and attack them. You don't want T cells to attack your own body. You want to make sure T cells are immunocompetent enough to recognize self-antigens versus foreign antigens. So the thymus itself, that whole gland, is able to test all of your T cells and present those T cells with self-antigens. When you present a T cell with a self-antigen, this T cell shouldn't react to it. It's an antigen that's part of your own self. Why would you want your immune system to attack your own cells? So if a T cell binds to a self-antigen during that testing process and it reacts to the self-antigen, that T cell has just failed the test. So if a T cell that's in the thymus takes the test and it binds to a self-antigen and it reacts to it, that T cell has just failed the test. So what happens? Well, that whole process is called negative selection. And if a T cell fails negative selection, it has to be eliminated. It's eliminated through two processes: clonal deletion, where the T cell literally just dies and gets eaten by a macrophage, or a process called anergy. The T cell stays alive, but it remains unresponsive. Now, kind of a crazy percentage here. Only 2% of all of your T cells are actually immunocompetent enough to go out into the rest of your body. 98% of the T cells in the maturation process in the thymus failed the test and attempted to attack your own body. So imagine how effective this negative selection process is if we are clearing 98% of those T cells because they are failing the test and only allowing 2% of the cells to actually go out into your body. Now, lucky for you, this is the exact same process of B cells. A few small differences, and that's just where the B cell matures. B cells are produced in the bone marrow, and they stay in the bone marrow to mature, hence the letter B. B cells will again undergo maturation and immunocompetence, where the bone marrow will present it with self-antigens. And if a B cell responds to the self-antigen, it's considered not immunocompetent. You don't want a B cell to react to your own self-antigens and start attacking your own body. So if a B cell does fail the test, it goes through the same process of either anergy or clonal deletion, and it gets cleared from your system. All the ones that pass the test are considered self-tolerant B cells, and these guys will start dividing rapidly, and they'll go out into your body to places like your spleen and your lymph node to hang out until you have some sort of immune reaction. But, and this is the last piece of this little segment, T cells and B cells cannot recognize antigens on their own, so they require APCs. Now, these are the same, this is the same material that you have on your end of the PowerPoint lecture, but I just rearranged it a little bit to give me some room to draw. So here's your T cell. The T cell is going to have a receptor on it. Here's the bacteria, and here's the bacterial antigen. Now you might be thinking, well, simplest way, the T cell's receptor comes and binds to this antigen. Well, that's in a perfect world, but that's not exactly how this works. So this bacteria cannot present its antigen to the T cell. T cells are blind to antigens that are on bacteria or viruses. So what we have to do is we have these huge cells called APCs. And APCs are called Antigen-Presenting cells. This APC will come and engulf the bacteria with the antigen. And it's going to eat that bacteria. And I'm drawing it one more time. So the bacteria and antigen are engulfed into the APC. The bacteria gets eaten and digested. The antigen, though, gets pushed out to the membrane and presented itself. This APC goes around to your entire body presenting this antigen, saying, hey guys, look what I have, look what I have. This is an antigen that doesn't belong to us. And this APC goes to the T cell and allows the T cell to bind to the antigen to actually look at it. So T cells and B cells, they require APCs to engulf the bacteria, disintegrate the bacteria, take the antigen, place it on its surface, and present it to the T cell itself. Only in this way can a T cell actually bind to the antigen and say, hey, I don't know who this guy is. I don't recognize him. So he starts dividing and creating this huge immune reaction. So we'll talk a little bit about APCs, and then we'll go through that mechanism one more time from the beginning. So APCs, antigen-presenting cells. These are cells that engulf the pathogen, degrade the pathogen, but save the antigen to present on their own surface to the rest of your immune system. The most common one is a macrophage. And a macrophage is just a really large cell that does phagocytosis. And so far what you've learned about macrophages is that they eat things like a Pac-Man. A new piece of information for you is that macrophages display foreign antigens to the rest of your immune system. The second type of APC is called a dendritic cell. These are APCs that are mobile. These guys can move around, and they're found usually in mucous membranes, and they are able to, again, phagocytize a bacteria and present the antigen to the rest of your immune system. The last one is a reticular cell. This is a stationary APC, and the only place these are found is going to be in the thymus. So 3 different cells all act as APCs, and this is how they work. APCs, antigen-presenting cells, if you see it here. The APC engulfs the bacteria and presents the antigen right here. That green thing is an antigen, that pink thing is an antigen. It presents the antigen on these little arms that stick out. These purple arms are called MHC proteins, major histocompatibility complex proteins. Every APC has an MHC on top of it. It actually has multiple MHCs on top of them. And those MHCs are what bind to the antigen to be able to present it to cells like T cells. If you notice here, it fits like a perfect puzzle piece into the T cell antigen receptor. These are T cell antigen receptors. These are the receptors that bind to the MHC antigen complex to be able to look at the antigen and say, hey, I don't recognize this antigen, so I'm going to create an immune response against all cells that have this antigen specifically. Because remember, T cells and B cells are your specific immune response. The antigen binding receptor binds to the antigen in MHC in order to see the antigen. If it sees that it doesn't recognize that antigen or that it's a foreign antigen, it's going to create a specific immune response where it multiplies itself and differentiates into the type of T cell that hunts for any cells that are presenting that particular antigen. So they're very specific on who they're hunting for and attacking. And this is just a nice little cartoon illustration that shows you that whole process again. The whole purple cell is the APC. The APC will phagocytize a bacteria. The lysosome comes and degrades the bacteria. And the little piece of the antigen from the bacteria is taken and presented on the MHC protein. So I know this MHC protein looks a tad different than what's here, but it's just 2 different cartoons. They represent the same thing. A key word on this slide that I'd like you to underline is epitope. The epitope is a small piece of the antigen that is sufficient for signaling. So this APC takes a small piece of the antigen from the bacteria, binds it to the MHC to present it to T cells and B cells. We call that small piece an epitope. So this is where we are going to stop on this PowerPoint. We will pick up the rest of this PowerPoint in a few days, so look out for the next lecture.